Great. Okay, so we're very, very pleased today to have Dr. Anand Hardikar joining us from Western Sydney in Australia. He actually is the chair of physiology and a professor at the Western Sydney University School of Medicine, as I mentioned, in Sydney, Australia. He received a PhD for his work in pancreas regenerative biology and undernutrition from the National Center for Cell Science, NCCS. Pune, India, and the WHO Collaborating Center at UCL, Belgium. He trained in cellular molecular biology at the University of Pennsylvania School of Medicine in Philadelphia and at the NIDDK in Bethesda. He then started his group in 2006 at the NCCS India and migrated to Australia in 2009. Within Australia, he's also worked with the University of New South Wales, the O'Brien Institute, the University of Melbourne, and the University of Sydney before joining Western Sydney University in 2020. He's also a visiting professor with the Danish Diabetes and Endocrine Academy since 2016, and I will drop the recent paper into the chat in just a minute. The title of his talk today is Developing a microRNA-based risk score for predicting the outcome of T1D therapies. Welcome, Anand. Thank you so much for joining us, and I'm dropping the paper here now for everyone else if they want to take a quick peek.
Thanks a lot, Monica, and really appreciate this opportunity to share some of our work. So that's a Research Square version that's not yet published. The paper's under review. What I'm going to share today is some of the data that went into the revision. So what I'm going to share today is unpublished, but yes, I'm happy to talk about it and discuss more about how this can engage collaborative potentials later on. So that's basically a picture of some of the people in our group. And please follow or connect. These are some of the funding agencies that we have. So the work in our group is mainly based on type 1 and type 2 diabetes biomarkers. But we also work on insulin-producing cells, islet cell differentiation, as well as epigenetics. But for today's talk, I'm going to talk mainly about T1D biomarkers. And that's the lab group. So for those of you interested, please feel free to go and look at our website, which has more information on the different biomarkers that we analyze and the different infrastructure capacities and groups that we interact with. So this is something that I had to put for a T1D biomarker talk because right from Eisenbach's early presentation of progression to or progressive decline in functional beta cell mass, this is something that we can't miss. But what I would like to bring attention to is that progression to type 1 diabetes has been identified with two major biomarkers. One is the presence of 2 or more autoantibodies, and secondly, having a genetic risk. And these have been some of the really key biomarkers that have driven all of the research and the progress that has happened until today. However, I'm going to take a stance, and because Monica said this was a think tank presentation, it made me put in an analogy, which I thought would be good to discuss for this group. So I was thinking of this while me and Mikta were driving in the car, and we thought that the genetic risk score is very analogous to the safety rating on a car. Because if you have a car like this on the right-hand side, which has a low safety rating, it's equivalent to having a high risk of being in a fatal accident if you meet one on the road. But if you have a high safety rating on the car, it's equivalent to having a low risk. The important thing is that if you have, I mean, this is something that we can't decide because we are born with a genetic risk score, and genetic risk scores are the first ones that we can measure in our life. But we don't simply buy a car and keep it in a garage. We drive it, and driving or living is highly dynamic. So it's also dependent on where you drive, and your risk dependent not only on the different precipitating factors that one encounters in their life, but also on the kind of genetic risk score that you have. But again, the other thing that I wanted to you all to think about is driving is not only dynamic, but it's relative to the context. So it depends on ethnicity and environment. And so, for example, if I were to drive to Uluru, which is a place towards the center of Australia, I would probably not see many cars on the road, but see more kangaroos. Whereas if I was driving in Southeast Asia, the precipitating factors, for example, each of these cars is a precipitating factors, maybe the virus, environment, whatever, drugs. Those are different than the context we see somewhere else. And in the US, Monica, you would be driving on the right side of the road. Which is very different from what we drive in Australia. So we have to remember that the context is different. And so what we put together in here was that it's great to have a genetic risk score, but what we should also start thinking about more specifically is a dynamic risk score, one that changes with the environment. And so this is an example where you can see a high genetic risk. The GRS will remain constant over lifetime once you measure it in both the scenarios. But now we know that there are drugs that can modify the progression of T1D, like teplizumab. And so a dynamic risk score should actually show that the risk lowers after use of the drug with a dynamic risk score like this. Those of you who know WACOR, you would know that that was a rodenticide used in one of the states where high concentrations of WACOR were associated with progression to type 1 diabetes. And that was something that could be, so there can be factors that can lower the risk or increase the risk of progression to T1D. And for that, we need something that can dynamically change. So what are the biomarkers that can dynamically change? And thinking of those, one of the best example that has been recently published through the GPAD group was glucose. It's a biomarker that is dynamic. It changes with time. And we know that glucose can be used. Continuous glucose monitoring was used in this case to predict the progression to autoantibody positivity. So they were able to identify autoantibody progressors in the study, and that's great. So I want to just summarize this part of the talk, saying that there are several benefits to measuring these biomarkers, and one of them is that autoantibody biomarkers are dynamic. They can be used and have been used in population screening, but genetic risk scores, they are based on SNPs, but they have been mainly from an European ancestry. So that's one of the strengths as well as limitations. The limitation that I mentioned before was that they're hardwired to our genome. So you measure it earliest, that's the benefit, but they stay constant over the lifetime. The other thing that we have to remember about autoantibodies is we haven't been able to see a good number of these to be stably reversed once seroconverted. So what it brings me is to remember the Atkinson-Maniela's paper of streetlight effect. And are we really looking at a streetlight effect when we are assessing glucose or autoantibodies? Because these mark the asymptomatic phase of type 1 diabetes. And so this is more reason to start thinking of understanding and looking at other dynamic biomarkers. Now, the one that I want to speak about are microRNAs, and I mean, on the left-hand side, you can see how different microRNAs target different genes in pancreas development and also in insulin secretion. And a bit more recently, we published a paper showing that it is microRNAs that drive insulin transcription, not insulin transcription driving microRNAs. So that was something which was a causal thing that we published a few years back. Now, just to bring everyone on the same page, microRNAs were first discovered in 1993. We saw Victor Ambros and Gary Rookin receiving the Nobel Prize this year, which was great to see. They are short non-coding RNA molecules, which are post-transcriptional regulators. They're highly conserved across evolution. What is important to us really is that they can be reliably measured in serum, plasma, TS, urine, sputum, most of the biofluids that we have measured, and they can be reliably measured. They're also highly stable, so they're significantly resistant to freeze-thaw or other kind of degradation than other biomarkers that we measure. So what are the studies that have been carried out? And I'm happy to provide a table or a PRISM file on this plot, but just to put together in context, this is not something like the first study on microRNAs that we are publishing. There have been way many studies before that other labs have published. And that started from 2012, when we were just starting to identify microRNAs in circulation. But there have been studies which have been a discovery replication. So the discovery cohort, so the bubble size here is the number of individuals in that study, the participant number, as we have shown here. The blue dots here are basically the discovery cohort size. The green ones are validation. If the studies are connected by a line, then that's a paper that carried a discovery validation in their report. There have been several of these candidate reports, which have been just taken from the literature and testing it in the plasma or serum of individuals with T1D. What we are going to tell you about, I'm going to tell you about, is our study where we carried a discovery on around 254 samples in our lab, but also more than 2,500 dry lab samples for the discovery. And then we had a validation set shown in green of over 2,204 individuals. But we also looked at a replication, which was an independent validation in 662 individuals across 6 different countries. And we also had an application for this, which is looking at the use of this risk score in treatment therapies for T1D. So that's what I'm going to talk about today. Now, the method that we use for measuring microRNAs starts with getting de-identified plasma samples. We use around 100 microliters of plasma. We spike it with an Arabidopsis microRNA. This is a microRNA which is not found in humans. Then we take it for RNA isolation on a robotic system. We introduce a different spike-in microRNA. And these are state-specific spike-in microRNAs to account for any unintentional loss of sample during the processing. We then carry a Th1n-based real-time PCR on an OpenArray platform, which is a FDA-approved kind of system, equivalent system. The data is locked into a trackable database where our collaborators can see it and share it. And then we carry QC, and we do validation using at least 2 or preferably 3 different workflows in R and Python and whatnot. So we have a CV of under 5% on the OpenArray. So we are very happy with this platform. And we use a lot of controls. So this is what the panel looks like. And just to say that this is what a square here represents what one sample would be going on. So this is a selected set of the predict T1D microRNA panel that I'm speaking about. But then we have a lot of controls. We use intrinsic controls, which are loading controls. There are RNA spike-in controls that I mentioned, the RT spike-in control. There's housekeeping gene controls. There are negative controls, which are microRNAs, which are not detected in the human samples. There are duplicate microRNAs, like these 2 green ones, which are 2 different assays for microRNAs, but for the same microRNA. So we can see if the 2 different assays perform similarly. There are positional microRNAs, so the same microRNA assay, but 2 different positions in the panel. So that's an important control to have because you can see sometimes some positional differences. There are some random repeats, which are samples which we repeat across the panel. There are technical and batch controls, which we include on every panel. And we also look for hemolysis based on these set of microRNAs. So those are the set of controls that we recommend and use in our study. Now, in terms of the study design, this is something that, Monica, thanks for posting that, but that was a previous version. We couldn't upload this, but this is a manuscript currently under review and it's unpublished. These are some of the data that I'm sharing you from the recent version. But what was good for us to see was that even after increasing the number of samples, our predictions still hold. So that was something that I'm going to show you. Okay, so we had a discovery cohort of 254 biological samples, which mainly included 189 human tissues. And this was majority of them were human pancreatic cadaveric islet donors. So we had a focus on looking at islet-enriched microRNAs, but we also wanted to look at the microRNAs that are important during human pancreas development, because we know that when pancreatic islets are destroyed, the developmental or the regenerative pathways recapitulate the pancreatic developmental pathways. So we know that those microRNAs are really important from the work of Doug Merton and several others. And then we also wanted to look at some clinical samples taken from individuals at clinical diagnosis, so within the first 48 to 72 hours of T1D diagnosis. Now, because we had fewer samples with clinical diagnosis, we also looked at previous reports from 21 different published studies, and that was 2,583 study participants. And we confirmed that the microRNAs which we had were also selected in those studies. But we also included some microRNAs from the literature that did not meet statistical significance in our hands, but were statistical significant for others. We then used a multi-ethnic cohort, and for all reasons, because of environment and ethnicity and mixed races, I'm going to call it as a context. So we had a multi-context cohort from Australia, Denmark, Hong Kong, and India. I would just like to point out that we were looking at case and controls, but in Denmark, the controls were individuals who were siblings of cases of individuals with T1D. We use these in a train test kind of method to generate a dynamic risk score for the 4 different contexts, which we call as DRS4C. We then used generative AI, and I will explain this a bit, to generate an enhanced DRS4C signature. And then we tested that in an independent validation cohort of 662 individuals, and also in 2 different applications of this EDRS in an established type 1 diabetes therapy in islet transplantation, as well as an emerging T1D therapy, the imatinib trial. So that's the set of microRNAs. As I said, most of them come from tissue, basic tissue-producing islets, islet-enriched microRNAs. There are some that were identified, and there's quite a good overlap, as you can see, between the plasma and the tissue. There are some which were identified from developing pancreas, and then these are the ones from literature, the negative controls and everything. What was interesting to see that from the previously published reports until now, we see that several of them are associated with genetic risk. They are either associated with autoantibody, they are associated with early onset of T1D, and significantly different between T1D and controls. So the first thing that we wanted to do was to generate models using a random forest-based workflow, which was the most appropriate one. And we split the Australia, Denmark, Hong Kong, and India cohorts into a train set and a test set, just randomly splitting them. And then we looked at the model. So if we train and generate a risk score for Australia, Denmark, Hong Kong, and India independently, and test it on their own test cohort, then we get a very good AUC, right? Which is what we are seeing even with GRS. But then when we looked at combining this test set together and testing each of these single country risk scores on a combined test set, the AUCs was not performing best. So that was the major reason why we thought that we can't use a single country risk score. We need to have a combined multi-context risk score generated. So we generated this multi-context dynamic risk score on this cohort of 2,204 individuals. We had a reasonably good AUC of 0.78 with a F1 score of 0.81. And these were some of the most important microRNAs based on CHAP values for these plots. So what we wanted to do was then to look at ways to use generative AI and then test these in other cohorts. Now, just to give you an example, if these were the 4 different contexts, what we saw was the distribution of these microRNAs. So the green, let's say, are the controls and the red are cases. The distribution is a bit different of each of the microRNAs. But what is important to note is that there are several probabilities of expression that are not covered by the sample size, however big or small you have. So we use Gaussian copulas using the synthetic data vault to fill in synthetic samples that can cover under the same distributions, but it covers all of the remaining probabilities of gene expression. And then we used either a set of 1,000 synthetic samples. So the synthetic samples here are in gray. The colored ones are the actual samples in our T1D and control cohorts. And you can see that you can have 1,000 synthetic, 10,000 or 100,000 synthetic samples, and they all follow the same distribution. So we then trained the model using an augmented set, which was the 1,000 or 10,000 or 100,000 synthetic samples, along with the 1,543 real-life sample or the trained set samples. And when we used that and tested on an independent set of 662 individuals, we find that with increasing synthetic samples in our augmented data set, we had a better predictive power with a significantly good F1 score, as well as specificity and sensitivity. So now what we wanted to do was test if this could be used for prediction of future outcomes of T1D therapies. And one of the side that we know is islet transplantation has been a well-recognized T1D therapy in several countries, including Australia, Europe, Canada, and several other parts of the world. So what we had access through was samples from James Shapiro's group, where we had pre-transplant blood samples taken from the host. We had the post-transplant 1-hour samples and 24-hour samples, and we had outcomes at 1 month post-transplant. So we had outcomes at 6 months or 12 months as well, but these individuals have undergone another transplant between 1 and 6 months. So we didn't look at the 6-monthly outcome. But I'm going to show you data on the 1-month outcome. So the idea was to measure the EDRS4C at each of these time points, and then to predict if we can guess the outcome of the transplant accurately at 1 month. So we had 31 individuals who received a transplant. So these are longitudinal samples. We split them into 13 of them most of them had no requirement of exogenous insulin administration at 1 month, but some of them had, a few of them needed up to 0.1 units per kg per day at 1 month of transplant. Around 18 of them required much higher exogenous insulin requirement at 1 month. So we first looked at baseline clinical characteristics, and we were not able to see any baseline characteristics, including C-peptide, basal or stimulated, HbA1c, and other kind of indices individually or together. They were not able to separate the individuals who had low versus high exogenous insulin requirement at 1 month. But when we looked at the microRNAs using the EDRS4C model, we saw that not the pre or transplant, but the 1 hour post-transplant was able to very reliably classify them into responders and non-responders. And we had a better predictive power at 1 hour than 24 hours. So that was one of the findings that we had from this study. Now, the other thing that we wanted to do was to look at the imatinib trial. Now, those of you, most of you would know, the imatinib trial was carried by Steve Gittleman, and it was shown to retard or slow beta cell death for up to 12 months, was very sustained, and we look at the 12-month time point. Again, we looked at the initial baseline characteristics from clinical biochemical, so including age, weight, days of T1D, baseline glucose, baseline C-peptide, baseline insulin, the dose of insulin, but none of them independently or together in a model. We're able to stratify those who responded or not responded to imatinib. But when we use the microRNA-based risk scores, we are able to get a much better separation of the responders, which are, so just to give you an idea, so these are 17 of the microRNAs in the 50 panel of predict T1D microRNAs. And what we see is that 17 of them, all 17 of them are present at higher levels in those that were in the upper quartile of responsiveness to the therapy than those in the lower quartile. So again, just to give you a clear idea, these were 43 individuals who received imatinib for 6 months. The outcome was measured at 1 year. We looked at the lowest quartile of response at 1 year and selected 11 individuals of the 43 and the upper quartile and selected the top quartile individuals as well. And that's how they look in our combined dimensionality reduction algorithm. When we looked at this closely, we identified that MIR27B, which was the first or the highest ranked microRNA that separated them into responders and non-responders. And these are the other microRNAs in the decision tree that contributed to predicting who will respond. So these are all baseline measurement. So these are measurements from before the individuals went on the trial. And you can see again, these 4 microRNAs, as I showed you earlier, are significantly higher in those in the upper quartile. What was interesting even further was when we did pathway analysis, we found that imatinib, the drug used in this trial, targets 9 different genes, but these genes are very distinct than the 36 different genes that MIR27B, the first microRNA, targets in the pathway. So what we think is happening is there is a synergistic effect in the levels of microRNAs in the upper quartile individuals to the drug action. And so that's something that we think is quite interesting, because this is not an effect then that you can enhance by just increasing the drug dosage. But this is something that is a very good example of synergism that we see in terms of drug therapy. So my final data slide here is basically to tell you how this risk score can be then used by clinicians or in a single patient context. And so we thought, like, if there was one person just walking in your clinic and you're a clinician, what could this do? And there are several studies, like Maria Craig here follows, and there are several others who follow individuals, but this was one from the bigger cohort that Maria carried out. And this was an individual that Maria had seen from as early as 14 months of age here on the x-axis to 16 months when that individual was diagnosed with type 1 diabetes. And if we measure the EDR-S4C in that individual prospectively, then we found that they already had a higher risk of 0.6 probability of T1D when they were 14 months old. And that risk increased to 0.75 when they were 18 months old. We did not see any increase from the clinical data in terms of autoantibodies, but that was the earliest response of an existing high risk increasing at 18 months. So this is something that could be a earlier biomarker. This is the N of 1. But again, then if you were working on a clinical study, and these are siblings from individuals, siblings of individuals with type 1 diabetes. So all of these individuals are the Danish cohort, birth cohort, where Simran basically analyzed the genetic risk score, autoantibody, and the age, and we took together that model to stratify using a isolation forest algorithm. And the red dots that you see here are what the model identifies as outliers, or those individuals that are at highest risk of developing T1D. Now, these are samples which were taken around 12 years back, and we know that the ones in the red are the highest risk, but the ones that are labeled with the S number for the sibling, those are the individuals who progress to type 1 diabetes. What we see is that if we just took the top 10 features, then we could identify a similar number of progressors from this cohort as what would GRS or autoantibodies together identify. So we don't have any comparison on using GRS and microRNA risk score together, but that is something that we have been speaking with Richard, Oram, and others to try and see how we can use this together. So in short,
Mr. Anand, just quickly, can we just go
Yeah.
back to the imatinib trial? Laura Jacobson from University of Florida asked, what does microRNA look like in the placebo participants from the imatinib trial? Could the microRNA be identifying people who are likely to do well no matter what?
Right. So in the placebo, I don't have that plotted here, but when we plotted just the risk score in a longitudinal study relative to the placebo, the ones who had imatinib showed lowering of the risk. So we saw that the risk score changed as we plotted it relative to placebo. But what's really exciting for us is those that had higher levels of microRNAs were the ones who ended up being in the upper quartile of responsiveness to therapy. And that's where we think the synergism is really interesting, because these microRNAs target tyrosine kinase pathway genes that are not targeted by imatinib. So imatinib is a tyrosine kinase inhibitor, but these microRNAs also inhibit other genes in the tyrosine kinase pathway, and then those were the ones which were highly expressed at baseline. So even from the placebos, we could identify some individuals who could have responded better on imatinib. But that's something that's a study. So we now have interferon alpha from Christina Rother, exenatide, and we also have baricitinib from our friends in Melbourne. So the baricitinib study samples, we just recently received the approvals. So we're looking forward to analyze those. But we do see that certain drugs can, just using a certain drug can change the microRNA profile from baseline to end of the therapy. So we are going to use that. I mean, this is what I was going to summarize as well here, is we can use that for designing combination therapies for T1D. But in summary, what I was trying to, what I think I showed you was that we developed an enhanced dynamic risk score for microRNAs based on 4 different contexts, and we're able to see that it could be applied efficiently to other contexts as well. The predictive power of 4C signature was enhanced by using the generative AI approaches. The EDRS was validated in an independent set, as well as 2 different therapies for T1D. The microRNA-based dynamic risk score, as I showed you in the example for progressors to T1D changes, and we have been working through a project funded through Hemsley on developing a point of care to measure microRNAs in under 15 minutes. for under $5. So that's something that we are working on as well. But what we really think is important is the analysis of different drug treatments and the pathways that these microRNAs target would be really important in designing combination therapies for T1D. So with that, I would like to thank the people who did the work. Mukta, Wilson, Pooja, and Rishikesh were the key people, along with Vinod and Simran, who were involved from our Danish and other cohorts. Ryan and Riley are they are researchers with us, but they are also individuals with T1D. So that was a huge kind of input from them during the whole study. And thanks to all of the clinical collaborators and the study participants and the funding agencies, mainly Breakthrough T1D, JDRF Australia, and the NHMRC. Thank you.
Thank you very much, and that was fascinating. Really an interesting talk and approach, and extremely collaborative, which we always, you know, give a lot of accolades to. I wanted to open it up to other people. A lot of clapping in your audience here. Other scientists to ask questions. Please either unmute yourself or drop something in the chat. Welcome to have you take the time to exchange ideas and information.
Hey, this is Laura Jacobson. I really enjoyed it. Thank you so much. It was really clear and easy to follow, which I usually have a hard time with such complex methods, but that was great.
Thank you.
I would love to see the placebo data like plotted with your microRNA. I'm sure it's going to be in your paper that's coming out, so I'm excited to read that. Of interest, if you're interested, I do a lot with responders to immune therapies, but have not looked at microRNA before, mostly looking at like flow cytometry and immune cell subsets. So potentially would be interested in collaborating if you're interested for like future trials with anti-thymocyte globulin. Maybe we could look for your signature.
Yeah, absolutely. And look, we do have the T1D panels. Once the paper is out, the plan working with the company is to have that panel out as a product so that others can independently use it. But if you don't have the infrastructure, because we recommend certain instrumentation and it's all robotic, so most of the people are doing like loading it on the system and sipping coffee, but these are automated pathways that we learned are really important. So we are happy to analyze more samples. In terms of the placebos, we had it in a previous version which was taken out, but I'm just going to try and see if I have any. I mean, this was the so this was what I was saying, that relative to the placebos here, the risk kind of lowers and then goes up again, but we are seeing lowering of risk relative to placebos. But I don't. When we look at the other kind of microRNA biomarkers in the placebos, we can see, like, we have analyzed the interferon alpha, which was a positive outcome at one of the 2 doses. We do see that it changes the baseline microRNA. So I think, and we do collaborate with several people here who are working in the cancer field, where different immune checkpoint inhibitors are being trialed. But just looking at what changes the microRNA signature so that they can be better responders to a different drug. So that's how we are thinking it could help design combination therapies in T1D.
That's great. Thank
you. Thanks, Laura. Please feel free to email me, and I'm happy to follow up with any collaborative thoughts there. Yeah.
That's great. Anyone else? Feel free to unmute yourself. Great talk. Anand, with the host of proteins regulated by the microRNAs outside of the kinases, do you have plans to look at the protein levels in your cohorts? This is from Brenton Short.
Yeah, so thanks, Brenton. I expected that from you. But yes, as you know, we are measuring, the next step is I mean, microRNAs is our first love, but we like to look at every single thing. So we are looking at all other biomarkers that are dynamic, which includes cytokines, proteins, metabolites, lipids, and the whole idea is to try put all of this together to then enhance and enrich this prediction signature. Because, you know, we have to be right for something like T1D being prevalent in a few individuals. You have to be correct for 99% of the times, at least, to detect that small fraction of T1D individuals in the whole population. So we have Mukta running some of the cytokines in a few cohorts starting soon, and then we are following with proteomic biomarkers as well.
So yes, that's a really interesting question. I had a question for you regarding the microRNA repositories. Are there any community-built T1D specific microRNA repositories? We're very interested in sort of this whole idea of the community build of the repository. We initiated and facilitated the build of the T1D TCR and now BCR repository. It seems like it would be a really cool thing to have as you continue to build these signatures around these different populations.
Yeah, so yeah, absolutely. I mean, that is one of the things that we have been developing. So we started looking at the developing pancreas in, so if you look at our previous papers from 2006 to 2012, we were working on human pancreas development, looking at different microRNAs, but also looking at mouse regeneration. And then we started to develop islet enriched microRNA signatures. So I wouldn't call them specific because based on our experience, we don't see anything that is specific to the pancreas. We would say they are enriched in the pancreas, but not specific. But there can be a signature that it could be eventually called as specific to pancreas. But that's something that we think would be a multi-omic kind of thing rather than just microRNAs. But what we have been able to develop is our focus was mainly looking at what's in the islet, what comes out of developing pancreas, and what can we see in the plasma as well. So the whole focus was therefore seeing what happens at the site of action, and then what do we see as an effect of that in the plasma. So we just need to keep a broad view and select everything from there. So that's how the signature was designed. Okay.
Yeah, very exciting. You know, and of course, there's a whole host of uses for this type of signature. You know, as you mentioned that, you know, the tag teaming of different therapies, or even perhaps just early identification of someone who's on their way. So it's really exciting times for you and your lab and the whole collaborative group. Last call for questions from anyone. All right, then. Thank you again for speaking with us from Western Australia. We are excited to look for that paper, and please just give us a shout. And we, if it, you know, when it first gets out, we will be featuring it in our Week in Review, which comes out every Friday. All the T1D papers that are basically fit to be read are curated in our Week in Review. So thanks again. Yeah. We'll talk to you soon.
Thanks, Monica.
Yep.
Thanks for the opportunity, and great speaking with you. Thank you.
Great speaking with you. Have a great rest of your day. Bye-bye.